I start, I must bring your attention to the following statement, which contains important information relevant to my presentation. In summary, Royal Dutch Shell plc is a large organisation comprising a number of separate companies, so the use of the name Shell or group or words like we and us are used here as a convenient generalisation as appropriate. The second point refers to the fact that the information contains views about the future that are clearly subject to future risks and change, and we cannot accept liability for use of this information. Indeed, speaking as a member of the research team, my own perspective is clearly limited, so any explanations I give cannot be taken as a formal company view. Finally, you will note that, the presentation is marked copyright and may not be used further without permission. I thank you for your understanding. Thank you very much. It's certainly an honour to be speaking in this eminent establishment, and I'm delighted to have this opportunity to speak to you all here today. I guess being the last speaker of the day is a blessing and a curse, so if anybody feels themselves a little tired, I will understand, but I hope I can captivate your interest for a little longer. It also gives me a chance to look back actually on everything I've heard today, which I think has been very interesting. I've come here as much to learn, probably more to learn really than to contribute, because adapting something like synthetic biology to something of the scale of the global oil or energy industry is indeed a massive challenge. And I think one of the themes I've been constantly thinking about through this day is that challenge of scale up. We're going from genetics on the one side to something which I'll be talking about in a moment of global climate change. How do we actually start to get the scale to a point that one can make an impact on the other? Just in my title, actually, we use the word biodomain. We had to invent the word internally. Probably other people have used it. I invented it a few years ago because we were developing biofuels for quite a few years now within the company, but we recognized that the capabilities that we were developing were clearly going to be applicable throughout the rest of the industry in different ways, and that's a point I'm going to make shortly. So I did reflect a little bit around this. I'm trying on the one hand to generalize through the oil and energy industry, whilst at the same time it's very clear that every company, even within this industry, has a different approach. So I'll be speaking very much from a Shell perspective in terms of my illustrations. It is clear that biological systems are prevalent through industrial operations. They exist in subsurface microbial communities, in reservoirs. I mean, the ancient origins of crude oil are after all biological, and they remain active to date. It can also apply to the biodegradation, the need to deal with production water, etc. So right the way from there, right the way through to my main interest, which is putting biofuels into markets, we see a need to understand biological systems. The big problem for us, which I think is a contrast with everything else I've heard, perhaps with the possible exception of my previous speaker, is about scale. There's a big difference in coming up with something which is low volume, high value. At a pharmaceutical end, we're at the other end where it's large scale and extremely small margins. And anything may be very elegant, but unless it's actually going to be extremely cost effective, will face an enormous challenge in actually making a cut to be something that one would want to invest large scale in. The second thing I would have been reflecting upon is again in terms of scale, but this time it's scaling from biological and engineering systems at the genetic level, the actual processes of synthetic biology we've been discussing, up to the interface of large-scale biological systems and large-scale thermochemical processes. I'm in an industry, I wish I should have started with a large photograph of a super tanker, because I think it would indicate a little sense of both the scale and also the enormous inertia that we have in our industry. We have been designing refineries for 100 years, saying, oh, by the way, I want to put a biological process in. They said, well, just a minute, can you define the design criteria in exactly the same way, give me the same predictability, etc. And to some extent, one realizes that biological systems cannot be automatically dealt with in exactly the same way that we've been very comfortably designing thermochemical processes for many decades. And that really is, I think, a fascinating and important aspect of the relationship of synthetic biology to our industries, how bit by bit we can see a methodology and structure helping to build up to something which gives us confidence in really understanding the predictability of these processes if we're going to properly use them in engineering design. But we are clearly aware that there are enormous transformational changes going on in this area. I think it's an extremely exciting area that I am confident will play an increasing role in years to come. So I've mentioned the many applications that do exist in our industry, but I'm going to focus on biofuels because that's really our primary focus of attention at the moment. And Shell has been very interested in biofuels and very active in biofuels for a long time. It may be because we've actually been heavily involved in biological systems for a long time. We actually had a very major biological sciences laboratory, in fact, not too far from here at Sittingbourne, for many decades, and another one out at Modesto in California. Needless to say, we closed them down. But the good news for me was that many of the people remained in the company, and we were actually discovered we had a very rich vein of biological scientists, people with vast experience available to us when we started to pull this team together over the last 5 to 10 years. However, given the speed of change, the other thing that we've done is to look at open innovation as a method, not to recreate the hundreds of people laboratory that we had, but rather to look for a much more agile approach working with universities. And we have many one-to-one relationships with universities around the world in every continent. So I will skip quickly through this, but I think it is worth just pointing out the context of why are we into biofuels at all. The point is there's this great trilemma that we're all aware of. The step change in energy demand, which really comes from the enormous increase in world population. We've gone from 3 to 7 billion, in just a few decades. Will it top out at 9, 10 or more billion? We don't know. But what we do know is that not only will there be more people, but they will be expected not only to be fed, but to have even improved dietary requirements. They want greater mobility, and they will have greater energy demands. And history shows that there is a relationship between historic relationship between GDP and energy demand. Hopefully, with the last few years of recognizing energy efficiency, that will tail off a lot lower, hopefully somewhere down here rather than up there. But nevertheless, the challenge is enormous, and in order to provide the energy, we clearly also have to take into account the environmental stresses, in particular climate change associated with CO2 or CO2 equivalent emissions from agriculture. So transport accounts for about 23% of energy-related CO2 emissions, of which road transport is about 17%, the rest being aviation, marine and so on. And the main thing here is that we reckon that the number of vehicles on the road is likely to double to more than 2 billion by 2050. Although there will be electric vehicles, etc., that increase, because of the inertia I mentioned earlier, will still rely very, very heavily on liquid hydrocarbons. And that's been a key driver for biofuels. This is reflected in the positions of governments. Policies are really creating that market. More than 65 countries either have or are developing renewable fuel mandates. Certainly all the major markets, so that's most of the volume in the world. Most of the markets do have some form of biofuel mandate. The legislative priorities do differ, and it's quite true. And in my opinion, if you're concerned about climate change, almost fortuitous that maybe the concerns are over energy security, supporting domestic agriculture. There are many local issues which might actually have been a predominant one within a market, but fortunately they all also, if they drive towards biofuels and biofuels continue to provide a low carbon alternative, will indeed assist heavily in the environment. And so we can see again that biofuels could represent a significant proportion of the world's transport fuels in decades to come. So, just saying a little bit about ourselves and where that leaves us. We have actually been involved in biofuels for quite a few decades. We have been investing significantly in the infrastructure in order to ensure that those biofuels are developed and distributed, and our distribution of about 9 billion litres a year makes us probably the world's largest distributor of biofuels. So we are looking for a chain in bio which is comparable to the one we already have with fossil fuels. Where's it going to come from? Well, today, in order to meet those mandates, to meet the world demand, we start with what we call first generation, which are clearly which, of course, not only are built on existing agriculture, but also build on existing well-known conversion processes, hence the fermentation of via baker's yeast to ethanol being the predominant mechanism. Because we need enormous scale if we are actually going to impact on the CO2 demands of transport, we have to look very much at how can we get the scale and at the same time get the sustainability and acceptability of this process. So it's very important to us that there should be an adoption of standard processes in the world using well-to-wheel CO2 standards and consistent methods, so that ultimately biofuels are not just something which you make irrespective of its CO2 footprint, but actually there is some sort of incentivization or reward for coming up with more advanced biofuels with lower CO2. Otherwise, you're putting a lot more energy to come up with a better solution, but at the moment there is no mechanism to reward doing anything better than the minimum. Having said that, of those 65 mandates, we are now seeing step-by-step change from how much you make to what is the well-to-wheels, and whether it's explicit in terms of producing cellulosic ethanol or whether it's just a CO2 target will no doubt depend on individual markets. So the main thing really is that we need a level playing field. We work very hard with other bodies to ensure the sustainability, certification of sources of feedstocks, etc. I won't dwell on that now in view of time. One thing though, however, about time is this interesting graph here, which if we are going to become material, it's clear over decades that the total energy, this is a logarithmic scale, so if you want a new energy source that is going to become material, by that mean, say 1% of world's total energy, it can still take several decades. This is first generation biofuels. It's now probably around 3%, so we'd say that's significantly, it's definitely material on a global scale. We're now looking at advanced biofuels, which I think is this blue curve here, and our current estimates are, yeah, we've got a little way to go before you really start to seeing significant plants, but they're going to have to grow on a faster scale, be rolled out faster than any energy vector we've seen in the last few decades. So what are we doing to accelerate this? Well, the first thing is you need to get a platform, a first-generation platform, upon which you can develop advanced biofuels. We have announced a proposed joint venture with a company called Cosan in Brazil. This will allow us to put together a complete supply chain from sugarcane, which is the lowest CO2 and most sustainable source of biomass available in the world. It will be big. The total venture will be about $12 billion value. It will initially produce about 2 billion litres of ethanol a year, but we anticipate we can significantly grow that in future years. But as I said before, applying all the robust sustainability principles, standards that we would apply anywhere in the world. Moving forward then, first generation, how do we get to advanced? And the key thing is, can you now start using all those parts of the plants, whether it's the bagasse from the sugarcane, the stalks from the corn, wheat straw, etc. Then you do need enzymes to start breaking that down to produce some form of sugars which you can then ferment on to, say, ethanol in this case. We've been working with a company called Iogen for nearly a decade now in a joint program. We did actually get the world's first ethanol plant, cellulosic ethanol plant, operating in the year 2004. We were up to half a million litres in 2009. And the attraction of that is we've learned an enormous amount about medium-scale production. We've been able to do this, as you can see, for 7 years. So why haven't we rolled it out? We haven't rolled it out because, as we've heard before, it's the engineering process of, well, yes, you could do it now, but if you developed it a bit further, you could do it more cost effectively. And you just have, someone has to make a point at which you say, right, we're continuing to prove our understanding of what we could do. What point do you press the button? Well, I'll leave that to my colleagues in the company to say exactly when that might be. But you can see from a technological point of view, it's exactly in the area that we've been talking about. The good news is, if we look at the synthetic biology theme of today, I won't dwell on this because it's well known to you all. I certainly am very enthused by what I see in terms of the last 5 years of or more, well, really since the decade since the human genome, where the cost and speed of sequencing has dropped by several orders of magnitude. High throughput screening is now remarkably fast, not only just robotics, but we have things like fax machines that can screen single cells, 10 million cells an hour, whatever. But you can really start, so it becomes very data intensive, but of course that's the level at which you can really then start to move from empiricism into something which has some sort of structure and methodology and direction. Certainly the data, the open source data and so on, these are all critical to everyone. It's a level which will allow us, I think, collectively to move forward in this area. However, to date in our industry, we're still heavily reliant on statistically based selection processes such as directed evolution. And that brings me on to another partnership we've had for a number of years with a company called Codexis, who focus very much on developing enzymes through their own approach of directed evolution, stressing them against the various design parameters that we need in order to operate at large scale. And we now have a very close three-way collaboration between ourselves and Iogen, who I mentioned earlier, and Codexis, in order to get to that point when we think, right, we've got a really effective system that will be suitable for world-scale plant. So what else is in the pipeline? Well, let me just mention one thing as an example. Now, this is working with another partnership, Virant. In this case, it's a thermochemical process, but we can take the cellulosically derived sugars from the other process, so in a sort of very large, sort of hand-waving modular approach, you can see that we can then provide feedstocks for other processes in addition to cellulosic ethanol. In this case, by going to directly to hydrocarbons, you produce something which is totally fungible, can be put in at any concentration you like. You don't need separate storage facilities, blending, etc. Takes you straight through to a fully blendable gasoline component. So bio front end, thermochemical middle. The question is, okay, that's one way of doing it. It's available technology. Are there other biological systems and approaches which could do similar? So those are the sorts of areas that I think are interesting and challenging to us at present. So I will now try and summarize this. I'll summarize a few things that I glossed over, so this will be the first time I say some of them, but never mind. The first thing is that although I'm professing a good story around biofuels, there's no way we're saying it's a solution to the world's climate change problems. What we are saying is that they're one of a number of many things that have to be done in order to address this, whether it's improved engines efficiency, certain number of electric vehicles, gas, rather, etc., etc. So we see a wide range of technologies will all be required. We do focus on biofuels at the moment. They are the most realistic near-term commercial solution. The infrastructure in this industry is massive. You can't just put in a new technology without saying, right, whatever it is, it's going to be there for the next 50 years. So you really have to be pretty clear where you're going. With the vehicles, with the infrastructure, with the familiarity and the energy density of liquid hydrocarbons, we believe that they will remain a very significant part of the transport energy mix for many decades to come. We are really dependent on government policies and incentives to accelerate the development of more advanced biofuels. It is taking a lot of investment, and we've heard some numbers earlier today. I was smiling. Yes, I know the feeling, and we need the right market environment to justify those long-term investments to actually move up to the next stage in advanced biofuels. And I think just dropping to the end there in view of time, reflecting again back to the theme of today, I think understanding biological systems is important, not only for the biofuels, but for a wide range of potential applications through our industry. I think our methodologies will remain predominantly empirical for some time to come, but I'm very excited by the deeper understanding of fundamental behaviour that we're clearly seeing and learning from synthetic biology, and I look forward to some exciting direct applications in parts of our industry and hopefully in the near future. Thank you.
Okay, again, two very interesting presentations. Can we take a few questions?
François Kepes from Genopole in France. Question about biofuels. Something that apparently you didn't talk too much about the lipids as possible platforms, or let's say molecular platforms for biofuels. So is there any reason for that? Is it a choice, or is it simply a technology that will not be mature before the other technologies that are now that you are now contemplating? So my question is in brief is how about lipids as a molecular platform for biofuels?
No, that is a very good question. Yes, I left it out primarily in the interest of the short time I had to speak. I can talk for too long otherwise. Without a doubt, you just look at today's volumes. Over 90% of biofuels in the world are derived from sugar-based origins. And to date, if you rely on vegetable oils, they are not very efficient per hectare. So we're rather dependent on some fairly significant improvements in yield upstream in the feedstock if you're going to rely on plant-derived lipids. Clearly there are other very interesting areas such as algae and so on, but and we've certainly been very active in that area. But in terms of what's coming through the pipeline, we see those further down the funnel, if you like. So it's a more question of timing at this stage. And I say the reality that the moment lipids, vegetable oils, they may suit if you're in Germany and you can grow a lot of rapeseed, but it's not a global solution. Thank you.
It's a question at the back.
Ben Davis, University of Oxford. You made this comment in passing about Sittingbourne and your other site, and you said, and we closed them down, of course. I wonder if you might comment on that in the context of foresight and hindsight and planning.
Did you sense a certain sort of gritting of teeth? It's always the way. No, I think what it does say is how incredibly the world has changed in 15 to 20 years. They weren't obviously designed for this. They were in agrochemicals and other areas, and an industry of our size is constantly exploring areas and then rationalizing back to its core. So it's a natural cycle. I could quote 10 other such instances in different areas, but it just so happened that it was relevant to this talk, because in fact the good news is that much of the experts and expertise remained near at hand. So actually I see that as a benefit. But yes, the world has changed a lot. I mean, okay, I know there was the Rio and Kyoto in the past, but in fact my personal view is that climate change hasn't really hit the sort of public imagination and political imagination until about 5 years ago. It was really sort of 2006, IPCC 4. Perhaps I have a personal theory that the introduction of the word carbon footprint around 2006 has also captured people's imaginations. So that's history.
Luke.
Luke Alfie from Codexis for Royal Bourbon. I'm very interested in public attitudes and responses to this kind of technology, and so I was struck by your comment that even within your own company, different divisions or different parts of the company have very different responses to synthetic biology and the terminology of synthetic biology. So I wonder if you could expand on that.
Well, in the context of all those developments, we did also our surveys of looking at all the position papers, for instance, that recently were being published in different countries. We talked about, let's say, the high publicity type of Greg Venter publications and all the news around it. I get questions from the management within the company. What are we doing? Do we anything about this? Then you start talking to the different businesses. In fact, we revisit our position papers about that. And when you start doing this, you sense immediately that depending on where you are in a business value chain, that it's more or less appreciated if you profile this novel type of development. So, as mentioned before, in food and nutrition, that is not really appreciated. The association, and it also depends a little bit where you are in the world. That is, as also explained, that the company is growing its business now in a more global environment, and dependent where you are, you have different appreciations and ideas about the concepts, the wordings, and how to do it, and also across the various business fields. So it's also a little bit a dilemma. I think we discussed it also with, I mentioned this with Drew, for instance, before in one of the other meetings. At one point in time, you would like to make your point and profile a novel development that has to be recognized in a science community and broader. And at the same time, you also have to connect and blend in existing infrastructures and know-how infrastructure. So that is a difficult dilemma sometimes. It's what it is. I even experience myself, if I come back from certain meetings, that I have to talk and explain to one of my colleagues or a broader group of colleagues. We have to educate also our scientists and technicians to get used to novel concepts and wordings. And it takes time.
Okay, Paul.
Could I just follow up on that particular point?
Sorry, could you just...
Sorry, Paul Fremont, Imperial College. What was very interesting about your presentation, which I thought was really lovely, was the idea of this time that you had in terms of getting processes done in certain the metabolic engineering. But it seemed to me that you were actually achieving enormous speed, if you like, for adopting new industrial processes, at least in the succinic acid exemplar you gave, and I'm just wondering, do you think that you need synthetic biology to improve the efficiency of what you're already doing? That's a sort of devil's advocate question, because it seems to me that you're making enormous strides already.
Yeah, my answer to that is a simple yes. I think
Could you then say in what way it's going to make a difference?
As I try to address or to explain, I think in the end we still do a poor job in, and I recognize this also in your presentation about predictability and scale-ups and robustness. So in other talks, I think it was also mentioned you make monuments in that type of efforts you do, multi-year, multi-people efforts, significant money involved, and still the learnings are there. That is what I try to show you with the timelines, that you learn from every project you do. Nevertheless, I think we can still make enormous progress taking, let's say, the concepts and ideas that was here presented this day over a broad range of applications. I just addressed the bioprocess part, but there are many others.
Okay, Jeff.
Jeff Baldwin, Imperial College. Lionel, just a question for you. I guess it was, interesting to hear your presentation, and
I think you're Lionel. Yeah, that's me.
Perhaps
just checking who's who here. Sorry.
A different perspective on the use of cellulosic feedstock from what we heard from Kirsten at TMO Renewables, and we hear a lot of, I guess, kind of hype around cellulosic as an alternative feedstock, and I just wondered from your large-scale industrial perspective, how viable do you see that going forward? What will it take to make that, really the better viable alternative in terms of the sort of starch feedstocks?
Clearly, if you have to do more processing, it's going to be, a more difficult and expensive process. So, to some extent, one is also sort of waiting for the market environment. What is the point? It's a bit like, at home when you buy your next, upgraded computer. If you wait another year, it'll be better, but now's the right time. So I think there are commercial market decisions as to that. And it's also a question of scale and interest. So there are, we're not alone in developing cellulosic ethanol processes. There are other companies that are driving ahead also, and each will make its own decisions. So it's a question of sort of fine balance of all the technical and commercial issues, and how you as a company decide to answer that, I think.
Peter Simpson, Parliamentary and Scientific Committee. I have a question for Lionel. I was very surprised to see that on your map of South America, you appear to require the whole of the Amazon basin.
Yeah, well, thank you for raising that, because it's an opportunity to clarify. Yes, I have a much better map, but that wasn't the one that I was given here, which indicates that sugar cane plantations are only in the very far south, 2,000 kilometers south of the Amazon basin. Indeed, it's more of a temperate crop, would not actually grow well in the Amazon. So that's one point. But in fact, sugarcane, again, people worry, sugarcane, Brazil, Amazon. It is a very big country. Sugarcane itself is grown over, I think, is it 1.2% of the agricultural land? It's a very small proportion around Sao Paulo, which in turn was historically developed, so I'm told by our Brazilian colleagues, because it was near the Port of Santos a century ago. It has nothing at all. It's like worrying about olives in Finland. I think those are the sort of geographical distances that we're comparing here.
It just remains for me to ask us if we can thank the speakers again for some very interesting talks.
I'm not sure whether I ought to thank the person who volunteered my my role at this point of trying to sum up not only today but what you might get tomorrow. So what I thought I would do is just give us a slightly personal perspective on it. It seems to me that today was as interesting for the themes that were strongly articulated as for one or two themes that one might have expected to hear about and didn't. I mean, it's perfectly clear that George Post gave us a flying start and reminded us of the breadth and the potential reach of what we can call synthetic or design biology. And I think that it could be said that if you're looking for a unifying theme for all of us, it's that synthetic biology implies a goal, a stated goal that you then cannot avoid but must try to reach, rather than just saying what happens if I take this bit out of the biological system, what will happen. So it's a much more restrictive and demanding science. And it seemed to me that as we went on, we actually saw with a succession of speakers that partnership was a very overt theme, whether or not the person speaking had resources to die for. I mean, we certainly heard from our Chinese colleagues that if you think big and you spend big, you get big. And on the other hand, I've never been quite so reassured about the release of genetically modified creatures as with Luke Alfie's talk. I thought it was a model of good sense and awareness of the issues that are raised. And that's going to come back tomorrow because, as I'll say in a moment, it's going to be a theme, how we enable further rapid progress on several fronts. And it seems to me that the policy people in the societies who were basically responsible for putting together the structure of this first day did get it right, because we heard from people who've struggled perhaps for quite some time to put into practice the technologies we've been talking about. We heard from people who also had perhaps come in rather later, but by sheer determination over quite short periods had done something quite dramatic. And I would like to highlight that Guoping Zhao's relatively short space, and it was, for me, I'm slightly more familiar with the work he described, it was a firework display of achievement in synthetic biology. And I could have highlighted one or two other speakers as well. Modestly put, fantastic achievement. And so I think that we got a lot more reality than rhetoric, which is only right, I think. And it was also heartening that we heard from people interested in really large-scale efforts. And I mean, there's so much hot air talked about biofuels, and sometimes one feels an attempt to capture a public agenda rather than to get things done. It seems to me that we heard a lot of substance today that we can take away and reflect on. So what are the things that didn't get said? It seemed to me that we shouldn't, we cannot overestimate the biological importance and the importance for us of clonal selection and of directed evolution, the words we used. But I really think we must stress that for example, the work of Jason Chin, who couldn't be here today, he makes advances in expanding the genetic code by putting on the system the demand that it survive only if it optimizes that orthogonal process. And that's another way of making sure that your constructs work, so that although we heard quite properly about how you quantitatively interrogate systems, both model and advanced, I think we still need to couple that to the useful, slightly slipshod, probably not very quantitative biological idea of clonal selection, and you don't know about the things you throw away, but you get the thing that does what you want. And so to go to tomorrow, it seems to me we've got several sessions that promise much. We all want to know the enabling environment for research. What do researchers need in order to get the science done? We all want to know how to innovate, and we've got a strong group there. And then finally, and certainly not least, we're going to hear about an enabling environment for the users of this technology, I nearly said civilian onlookers, but you know what I mean. Those people who are the wider beneficiaries of what we're talking about. So I've been tremendously heartened. I'm also heartened by the rigor with which everybody tried to keep to time. I feel that you've earned your drink. And it only remains for me to point out to you that this time the doors will open at the back onto some refreshment, and to thank you all for coming and for taking an active part, and hope to see you all tomorrow.